The Science of Cancer, Part 4, the transformation from normal cells to cancer cells. Normal cells. The cells are made up by at least 800 to 1000 proteins and enzymes, as well as some hormones that make part of their inter and outer cell communication system known as cellular communication. The signals that stimulate the cell's basic functions. Controlled cell division, cell differentiation, cell mobility, death of a cell or apoptosis. Three abnormal situations that can turn a normal cell into a tumor cell. Excess of carcinogens or signal inductors. Mutation and amplification in the number of genes that produce cell receptors. Mutation and rise or reduction of genes that produce proteins that signal commands to cells. The excess of sign inductors or of carcinogens. TGF-beta, WNT, carcinogens. The excess of inductors or cells contaminated by carcinogens speed up cell signals uncontrollably. And the consequences are rise in cell division, changes in cell differentiation, allows the tumor cells to become mobile, the rise in gene mutations that produce cell receptors. Examples of the rise in genes that produce cell. GCPR receptors with G protein. These are large transmembrane receptors, and due to this, are part of 40% of modern treatment that blocks RAS inductors and signals. Main tumors associated: ovary, thyroid, colorectal, Kaposi sarcoma, prostate, gastrointestinal, and others. EGFR receptor of factors of epidermal growth. Out of the 9 different types of these receptors, HER2 is associated with aggressive breast cancer. Consequences of the rise in receptors: rise in cell division, changes in cell differentiation, allows the tumor cells to become mobile, starts neoangiogenesis, and inhibits apoptosis. The rise or reduction and mutation of genes that produce proteins that signal command to cells. In normal cells, that is a balance in the activities of oncogenes and gene suppressors that stop the transformation of normal cells into tumors. Cells that suffered mutations in one or more oncogene can become abnormal and cause tumors. These are the associated tumours. These imbalanced activities between oncogenes and gene suppressors cause uncontrollable cellular activity characterized by rise in cell division, changes in cell differentiation, allows the tumor cells to become mobile, starts neoangiogenesis, and inhibits apoptosis. In cases in which there is a lack or no action by the gene suppressor before an oncogene is raised, the imbalance can also cause a normal cell to turn into a tumor cell. Well-known gene suppressors are P53, around 50% of tumors are related to this, BRCA12, 5 to 10% of breast cancers, TGF, 75 are To resume, the transformation of normal cells to cancer cells shows these characteristics. Uncontrollable cell division, abnormal, incomplete, or non-existent cell differentiation, incontrollable cell mobility causing metastasis, cellular immortality or anti-apoptosis.